welcome to MBIOS Research Insider. Before we get started, I'm Eugene and I'm your host for today. So just a little housekeeping before we get started. So as you know that we are on live right now on Facebook, so please feel free to share our Facebook page, our live stream to your friends as well as your family members who actually wanted to join us. And also do feel free to actually, put some questions, raise your questions in the Facebook page as well. So yeah, so before we get started, if you do have any questions during the session, please type them into the question box in the chat features provided, or you may bring them up during the Q&A session at the end of the sharing session by using the raise hand feature. So now I would like to introduce our speaker, Mr. JianHua Lim. Mr. JianHua Lim is pursuing his Bioengineering Ph.D. at the University of Pennsylvania, and he is a recent graduate from the University of California, Berkeley, where he received his Bachelor of Science in Bioengineering. He is eager to pioneer research in single-cell analysis and profiling using novel BioMEMS, bio-microelectromechanical systems devices. His research experiences includes BioMEMS, genomics, bioinformatics, and immunology. His current research interests lie in combining these disciplines to develop point-of-care diagnostics, biosensors, and medical devices for disease diagnosis, prognosis, and treatments. He aspires to be a successful biotechnology entrepreneur and perform exciting research, discovering new ideas and solutions and integrating them to produce advanced products and services. So besides, he also obtained a certificate in entrepreneurship and technology from the Sutardja Center for Entrepreneurship and Technology, University of California, Berkeley. JianHua's ultimate goal is to advance the field of immunology and pathology on a global scale through biotech product commercialization while living a balanced lifestyle by traveling, engaging in sports, and enjoying food and music. So now, without further ado, we will return the time over to JianHua. So JianHua, the floor is yours.
All right, thank you so much. I hope everyone's doing well. Just let me, if you can allow me to share my screen real quick. All right, you guys can see my screen, good?
Yes, we can.
All right, that's awesome. So, hi everyone, my name is JianHua. Thank you for that wonderful introduction. Yeah, so today I'll be just giving a quick talk about what is this like to be a bioengineering PhD student in the US. So without further ado, I'm just gonna get right into it. Just a quick disclaimer. Yeah, so the talk I'm gonna give is just gonna contain some of my background, my education journey, how did I come into bioengineering, and then I'm going to talk about my current research work here at UPenn. And then my career prospects, and also just some general advice for those who is trying to apply for a PhD as well in the US. Yeah, so let's get into my background really quickly. So it started in 2017. I did my A-levels in Sunway, where I took biochem, physics, and math. And then right after, I applied to US universities and got accepted to Berkeley, a University of California, Berkeley, where I did my bioengineering undergraduate with a minor in electrical engineering, computer science, and also pursued a certificate for entrepreneurship and technology. And then currently, this is where I'm at right now. I'm a first-year PhD bioengineering student at University of Pennsylvania, where I'm doing my lab rotations. And essentially, lab rotations are where you get to give a flavor and taste of different labs before you actually commit to one for the next 5 years. So just going in really briefly about what is bioengineering so that we're all on the same page. Bioengineering is a multidisciplinary field that leverages on different techniques and disciplines from different traditional fields such as mechanical engineering, electrical engineering, chemical engineering, computer science, math, biochemistry, and you can see many others. With that being said, that the space of the bioengineering is just so vast that it's really hard to navigate. And even for me at the beginning, just because there's so many outlooks here. So to help us categorize this massive field of bioengineering, the general consensus, it's split into 4 different bioengineering concentrations, each with its own unique disciplines and fundamentals. So for the first bioengineering concentration, we have the cell and tissue engineering. This is really popular for creating engineered replacements for damaged tissues through the design of biocompatible materials. and also just to understand the cellular and physiological mechanics to construct engineered like scaffolding for cells. And then some kind of like the examples include like stem cells for tissue repair and replacement, anti-aging research, drug delivery, biomaterials for like scaffolding, cellular mechanobiology as well. And then secondly, we have biomedical devices, and this is kind of where my space is at. And so we kind of like focus on the development of new biomedical technology for life science research, and also trying to advance the healthcare using fundamentals from mechanical engineering and electrical engineering. Some kind of like examples include instrumentation, building prosthetics, and then the field of BioMEMS, which I will kind of elaborate on later. The field of biocommunications, biomicrofluidics, micro robots, and also just like wearables. And then we have the field of synthetic and computational biology, which kind of focuses on designing and building novel biological functions and system by kind of like applying engineering design principles and computation tools to biology to produce materials more cheaply and sustainably. And basically to design this kind of construct better by performing genetic systems quickly, reliably, and also safely. And kind of like some examples in this field include working on bio-inspired materials, working on surgical adhesives, using microorganisms to synthesize materials of medical or industrial value, such as like bioenergy, and also just like computational genetic systems to kind of re-engineer bacteria to produce what we want, or kind of like fight diseases. And then the last bioengineering concentration that I have for you is biomedical imaging, which kind of focuses on developing technology and applications for life science research, and also trying to advance the medical imaging system as a whole. So kind of some examples that include under this umbrella includes different medical imaging system, targeted radiotherapy, developing different smart contrast reagent, targeted molecular and cellular imaging, and developing like what we call a microscopic ruler. So you can see that just these four concentrations, they can be really highly interdisciplinary. And these concentrations are essentially just a set of guides that can really overlap. As promised, I'm just going to talk a little bit about BioMEMS, or Bio-MicroElectrical mechanical system. So this is basically where my research area kind of focused down into. And essentially what it is just a microsystem in the microscale, which is inspired by a transistor, which is MEMS. So instead of using electrical current in what you see in your phones, in your transistors, we are just leveraging on using water current to kind of play around with different tools. And the reason why I like it so much is just because it combines so many different bioengineering aspects. It combines like the application of the bio side. It combines the mechanical system, the electrical engineering to kind of make it digital available. So kind of giving an example of like what the first BioMEMS kind of device looked like is essentially the question of like, how do we even weigh a single cell? And researchers just thought about, why not let's just create a weighing scale that is the size comparable to a single cell. So that's the birth of BioMEMS here. Basically uses like mechanical, electrical, optical parameters to kind of like weigh that single cell. So that deflection would translate into the weight. And then we also have a big thing in BioMEMS, the diagnostics component, where we kind of work on, like, for example, PCR chips, DNA microarray, and also just various types of point of care diagnostic chips as well. Then we also have different tissue engineering applications for BioMEMS. So kind of working on what we call an organ on a chip, which is essentially just building, creating tissue constructs on top of devices. And what we can use that for is kind of like to automate cell culture, or even perform like drug testing, so that in the future we can avoid animal models. And then we have also medical implant surgery in the space of BioMEMS. This includes like implantable microelectrodes that kind of like measure the brain electrical impulses, develop micro tubes or robots for surgery, and also just different drug delivery systems using simple Palmer valve techniques. Yeah, so from my past research experience, especially during my undergrad, I've immersed myself in various research fields in bioengineering, and I really enjoy them because they are so highly interdisciplinary, and I can utilize the different distinct skill sets from each field. So yeah, I'm just going to talk a little bit about the different research experience that I have. As expected, I did some work in the field of BioMEMS. where I kind of worked on developing an on-chip combined single-cell imaging and also RNA sequencing on the same device here. I could work a little in the field of immunology, where I was using bioinformatics techniques to find kind of de novo protein-based vaccine constructs for SARS-CoV-2 and dengue virus. And this was actually done back in Sunway when I came back for a summer break. And then I also kind of used computational biology to work on extracting and analyzing cellular mechanics data from this organ on chip called what we call a spine on a chip. Essentially, it's just like a device with spine cells being cultured on it, and it's just being stretched to expose itself to various stretching cycles. I also did some bioinformatics research in the field of genomics, where I was doing research on how plant pathogens kind of modulate microRNA silencing of the plant's innate immune receptor genes. Finally, I worked on developing a rapid point-of-care diagnostic device for SARS-CoV-2 using a novel microbubbling readout method. And this was actually my first research rotation project here at UPenn before I kind of dive into my current research work, which is in my second research rotation here. So I have two projects. The first one is a ultra-fast multiplex cyclic imaging. And essentially what it is giving some background for the biology side of application. We want to kind of like understand the dynamics of protein biomarkers from tumor cells so that we can actually perform diagnostics and also just monitor cancer treatment over a period of time. So if you think about like a bench to bedside scenario, one can kind of like isolate, for instance, the circulating tumor cells from liquid biopsy of patients to understand the temporal dynamics of specific protein biomarkers for early diagnostics, or even just like drug efficacy monitoring. And then using those data, we can then feed them back into reevaluating diagnostics methods and also just like better informing us of the drug choice and also just the drug dosage. However, there's still a need for a rapid and accurate method to quantitate these kind of biomarkers, especially both spatially and also like temporally as well. So to do that, we kind of look into this microfluidic system, which is called CODEX, or code detection by indexing. Essentially, it's just an automated microfluidic platform that allows the direct visualization and also just the quantitation of protein biomarkers. So this was actually developed and commercialized at Stanford University here in the US. And it has been demonstrated to simultaneously kind of visualize up to 60 different biomarkers in situ. Just going over the technology really briefly, CODEX performs kind of this what we call one-pot DNA conjugate antibody staining, essentially just like slapping all the antibody on top of tissue so we can actually like stain them in future runs. So we want to do that on like tissue samples, and then we would then follow by kind of like a multi-cycle reaction of kind of adding fluorescent labeled DNA probes. And then once you add them on, you can kind of image the probes, and then you strip the probes away for the next cycle, and you can just keep repeating it. And then once we are done with that, you can kind of collect all those images at the end, you stitch them up together and you stack them so that you can kind of like track the protein expression across time. The problem for this current technology is its lengthy cycling time of around 1 hour for each cycle to kind of just like strip off the DNA probes after imaging. So that means that if, let's say, I was to do like the 60 different biomarkers and have like 3 different colors, it takes like 20 cycles, which means that it takes 20 hours just to do like 60 protein biomarkers of imaging. So that's really bad because we have a lower throughput across the different cycles. And also just in case that we want to look at the temporal dynamics of protein expression, we're going to have that big gap in time gap that we won't be able to kind of like probe it. And therefore, there's just a lower granularity in those kind of temporal dynamics. So therefore, my project here was to hijack this component of this CODEX technology and also just further optimize it to create this next generation of CODEX technology. So the approach we're using here is just called a Scission-accelerated fluorophore exchange, or the SAFE technology. So it leverages on the rapid bioorthogonal click chemistry, which recently just won the Nobel Prize for, to kind of remove the immunofluorescent signals after you image them. And that allows us to rapidly kind of image over cycles. So in brief, this SAFE technology will quench the fluorescent signal, which essentially just turns it off, the fluorophore. Then after that, it completely removes the quench fluorophore by cleaving it off from the antibody-labeled cells or tissue at a very, very high speeds of around 1 minute. So the cycling time will essentially decrease from 1 hour to 1 minute if you can achieve that. So the way that it works in brief is using this cooperative bioorthogonal chemistry of tetrazine, Tz, and also a cleavable transcyclooctene Tz-TCO. And essentially the Tz here is linked to a quencher that will kind of quench the fluorophore that is bound to the Tz-TCO when it's in close proximity. And then with that, it will also cleave it off so that we can wash it away and just like recycle the probes as well. Yeah, just bringing this back into a larger bench picture. This is how we can actually like situate ourselves by introducing SAFE into CODEX. So if you can couple CODEX multiplexing and automated capability with SAFE's rapid bioorthogonal cycling of the fluorescent probe, the throughput of the combined technology can be just vastly improved that will further benefit cancer diagnostic and therapeutics in the long run. So just giving a little bit more a background like the reagent synthesis on like how we even like prepare all these different reagents. It's a lot of organic chemistry, and to be fair, it's really not up my alley, and this is like my first time working on organic chemistry reactions, but nonetheless, it's a really exciting field. So essentially we want to prepare antibody DNA and a complementary DNA fluorophore that will hybridize together. And we use techniques such as amide synthesis between amine groups and ester groups, which we use this ester group called NHS, and then also just using Tz and Tz-TCO click chemistry that I mentioned before to kind of like bring the two molecule complexes together. So basically here, the antibody DNA will be a one-pot stain reagent on the tissue samples, and then the DNA fluorophore probes will then hybridize to those stained antibody DNA for visualization purposes. So in terms of like antibody DNA conjugation, we will first prepare this antibody TCO complex by reacting the amine groups on the antibody with NHS TCO complex. And then we also want to prepare alongside with it the tcDNA by reacting these kind of like functionalized amine groups on the DNA marker with NHS Tz. And then finally, we will click them together to form this antibody DNA reagent. And then in terms of synthesizing the DNA fluorophore, it's really much more straightforward. Here we just straight up slap on this DNA with an amine group with NHS TCO fluorophore complex, and then it would just form like a DNA fluorophore. So just to show you like what its application is, we kind of like did antibody staining on like on this like surface protein called the EGFR protein on fixed cells. Here we use this A431 cancer cell line. You can see that the DNA fluorophore probes is basically hybridizing to its anti-EGFR antibody DNA counterparts. And then we also did some Hoysch nuclei staining. Essentially, it just like shows the nucleus of a cell, and also just to serve as a control to check that there's no spectral and spatial overlap between the two dyes here in the composite images here. And then next, we also kind of show the efficacy of like quenching the fluorophore. So basically just turning it off once we're done imaging. You can see in this image here, video here, we can see that it turns off after 15 seconds. That is exactly when we add the fluorophore. So basically my next step here is to kind of like broaden the antibody panel to test for different proteins, because this was only test on one. And even possible on just like RNA like products as well, so that we can just understand the different spatial regions of the cell, and also just to test the robustness of this technology here. Yeah, so that was my first project. And then just moving on to the second project where I did this paired loading and droplets, which I will just talk a little bit more. But before I dive into that, just giving some background in the bio side of things. Essentially, we want to understand like the immune response to tumors because it's just such a complex condition here. So immune cells can basically undergo this process called immunoediting, which is kind of inhibiting tumor growth and progression through the recognition and rejection of malignant cells here. However, immune responses can also promote tumor cell growth, survival, and also angiogenesis through the induction of oncogenic inflammation here. And then tumors can also generate kind of this like immunosuppressive microenvironment that can block productive anti-tumor immunity. And this is really a barrier, current barrier to effective immunotherapy right now. So if you can really understand this kind of a complicated relationship, we'll be able to generate like kind of protective anti-tumor responses in patients. The problem is that most immune-tumor relationships that are collected in bulk measurements, which essentially means that we have multiple immune cells and tumor cells in one measurement collection. And that's a problem because we then lose kind of like this heterogeneity in their relationship that can lead to missing out on potential key, but possibly like really rare signaling responses. So we need a way to kind of isolate kind of single immune cells and single tumor cells in their very own kind of like microenvironment here. And then using this like microenvironment can freely manipulate individual pairs and observe at the single cell level the kind of interactions such as their extracellular vesicle secretions here, which tells us a lot about their protein expression and just gene expression just in general. Yeah, and to do so, we leverage back on BioMEMS again. So we're using this field of BioMEMS called droplet microfluidics to kind of like capture single pairs of tumor in immune cells. This device is basically called a microfluidic particle zipper, basically to kind of like control loading of two cells into droplets. So usually in droplet microfluidics, we are limited by the statistical limitation called the Poisson's ratio, which tells us that if we don't pack the cells together or kind of like order them, we will get around, I'll say like 10% of droplets will have one cell and around like 1% of the droplets will have two cells inside it. So essentially by packing it, this device is able to kind of overcome this Poisson's ratio, kind of like packing them where they meet at the center, kind of resembling a zipper. That's why it's called a particle zipper here. In terms of our application, we're using live cells instead of hydrogels, which poses kind of two main differences here. First, the hydrogels can be really highly packed just because they are so deformable, and then cells are not as deformable. And even if you can pack the cells closer together and deform them, the shear stresses might cause like mechanical perturbations that could disrupt cell signaling. And that's not what we want because we want to keep it in the nascent stage. And then secondly, we also have like cellular secretions from live cell, which causes like aggregations when you start like packing them really closely together. And that just like kind of makes it hard to encapsulate a perfect pair of cells. But yeah, nonetheless, we kind of just use this as our springboard kind of a proof of concept and just like start optimizing from there. So you can see this like video here that shows how we kind of like stain two different live cells with two different colors, one in red, one in green. And then we just feed them into the device here. You can see in the video on the left, the cells just flowing into the zipper here. And then in the middle, you can see the droplets kind of forming that. It's a little hard to see here, but you can see like the cells being encapsulated inside it. The problem is that right now we see that on the picture on the right, there are just really few droplets that contain just one cell, let alone two cells that we really want to achieve. So I actually started this project about a month and a half ago. So there's still a lot of work to do into optimizing this. So there's definitely just more work that needs to be done in terms of like both the optimization and also like the device design as well. Yeah, so now you know who I am, what I do. Let's just shift the talk to more of like a broader outlook for everyone, and kind of like some of the career prospect as a bioengineer, and also just like the application process for PhD programs in the US. Yeah, so just giving a little background, the current employment in the US in 2021. You can see that most of the market is just dominated by research and development. And then also we have medical equipment, supplies manufacturing, and even all the way down to academia as well. So I like to break it down into this way on the right. So academia essentially includes research like such as postdoctoral fellows, and even going for professorship as well. And then we have another big segment that includes industry players, such as different kinds of engineers that could include, for example, like biomechanical engineer, bioelectrical engineer. We have people working in operations, project management, regulatory affairs, and also just being a consultant to biotech firms and whatnot as well. And then we also have the medical classes. A lot of bioengineers actually are interested in the medical scenes as well, and actually go and pursue their medical degree and come out to be physicians and also just like clinicians in general. And then also last but not least, my very own favorite, having this group of entrepreneurs stemming from bioengineers, just basically working and like forming startups, working in venture capital as well. Yeah, so we know about like what kind of like makeup for the job prospects are. We also want to know like how much it really pays out, you know, whether it's a field worth into like diving deep into. So you can see in 2021, the median wage in the US is 97,000, with the upper 10% earning 150,000 and the lower 10% of like 60,000. You can see that right now I'm currently in the academia scene as a PhD student. And yeah, so we basically like have a median wage of around like 60,000, which is not too far off from like other aspects, but it's definitely a place where we can definitely kind of push up in terms of like a job payment as well. So yeah, that leads us to the question of for people who are interested in applying to PhD program, is PhD a right pathway for you? And just kind of like thinking about the couple of months that I've started my PhD, what kind of like the skill sets that has really been like demanding out of me, includes like a couple of them as the following. So we have like really, you need to have really strong like intellectual curiosity. That doesn't mean that you have to be the smartest in the room, as long as you have that drive to keep on learning and just like challenging the status quo, kind of like working in novel research, and also just like pushing the forefront of what you're working on the research. That's something really important. And then we also have skill sets that require leadership and ownership. And that's because that you would only care most about your own projects. That's a blunt way to put it. No other people is going to care as much about the project as you would do it. And that's why that you really have to take initiative to kind of push for your projects, and also just making sure that your project has its right application and it's fit into the right market as well. And then we also need to have a lot of determination. PhD program is just filled with a lot of failures and hurdles. Even with my short process of like 3 months here, I actually hit a lot of roadblocks. I'll say I hit roadblocks probably like 80% of my time, even in the first like 3 months here. But nonetheless, those are what actually like makes you really push forward into novelty and your forefront of the research. So that's something that you have to keep in your mind for like the next 5 years. And then last but not least, like you have to have really good communication skills. And even if you don't really have good communication skills, like just like learning like how to effectively communicate to a diverse group of people is really important. So for instance, right now I'm communicating to different undergraduate students as well. But what if I'm trying to communicate my research to, let's say, high school students or like middle school students who are just like learning about very basic biology application? So that's really like different, and the way to go about that is to just be really effective in communicating. And then just giving like a brief application timeline of how PhD programs work in the US. So you start off the May of when you start applying. You want to start exploring different schools and programs, and also just really start identifying interested faculty, and start drafting your personal statement. And then if PhD programs require you to take standardized testing such as GRE, you can kind of like decide whether you want to take it, because most of the programs right now is really optional. My current opinion about this is that you only want to take the GRE only if it's really necessary. So if you can like not take it, like you don't have to take it just because it's a money sink. You're gonna pay a lot for your GRE examinations. And also it's just, we have a lot of this like ethical question that comes about when like different people have with different financial background. Some people will not be able to take it just because they lack of the funding, and that kind of like just puts them into different application weight classes. So, and to make it more like holistic, you just kind of like reduce that the aspect in applications. And then we also want to shortlist schools and programs in around like August. And then we also want to prepare for fellowship applications. And fellowships are essentially giving you extra money on top of your PhD program. So usually a PhD program, I'll talk about it more in the next slide, will fully cover your tuition and will pay for your insurance and give you like certain amount of stipend. So the fellowship is kind of just on top of that. And most fellowships are actually limited to a US citizen and permanent resident, but a couple of them that are really kind of highly regarded around the world, such as like Stanford's Knight Hennessy Scholarship that allows for anyone to just apply for that. And that's also the same time in August where you want to take your GRE, but only if it required, as like we discussed before this. In October, we kind of want to start preparing for graduate program applications. And also this is where your application deadline for your fellowships are going to start trickling in. And then you also want to kind of reach out to interested faculty and just say like, hey, I'm interested in applying to the PhD program in your school. I'm interested in working with you. Like, do you think like you have some time to talk about research? You think like your lab has like space, has like the required funding? Basically just like set up a connection with the faculty so that they can kind of piece together your application paperwork with your face when they read it later on. And then you also want to start asking for letter recommendations, which is really important. I will just talk a bit more later on. And December is where you apply for all your PhD programs. That's the deadline for most of them. And then in March, you start hearing back from different graduate programs. And for successful applications, you will start getting interviews into the next stage. Most of the interviews here are kind of just to check that if you're a real person, you are what you say, and more than often you're already into the PhD program. And so what they do is if you're within like the vicinity, they might even fly you out into their graduate school just to visit for over like a recruitment weekend. And then last but not least, you like make a decision and really celebrate. And then some kind of application tips here. So for the level of education to pursue a PhD program in the US, you do not need a master's to be accepted into a PhD program. So just like how I did it, I just applied at my last year of my bachelor's and just got accepted into a PhD program. In terms of like selecting a program, there's no such thing as backup school as like you expect in an undergraduate school selection, unless you're only just going in for the title of the PhD. And that's because when you're working on something for the 5 years, you're going to want to work on something that you're really interested in and not just because I'm just going for that title of a PhD. You also want to consider if the school offers rotation program. So for example, UPenn offers rotation program that allows me to kind of like select different labs, giving a taste of what it feels like to be in the different lab to understand like the lab culture, the kind of different projects they're working on, if it's a good fit for me, and also if I'm a good fit for them before kind of committing for the next 5 years. So if that's like a selection criteria, like you do need to look out for that. And so you also want to kind of choose program with at least 3 faculties in that program that you're interested in. If you're just going in just for the 1 faculty, that's a major red flag that you do not want to go just because like what if the faculty they're interested in working is going to retire soon, or it doesn't have the financial capacity to take you on. In terms of application competence, the quality of research experience is really a key admission factors. So a lot of people say that, oh, you have to have a really strong GPA. That is true for PhD programs because essentially it's testing your intellectual curiosity here. But I've seen a lot of people that does quality research work with lower GPA that really get into really top schools. So that goes to show that when you're doing your research, just really make sure to do like real quality and not just saying that, oh, you're just seeing the research experience, just putting in your resume. And then also in terms of like number of publications, that doesn't really equate to the application competence. I've seen people with like 4 different first order publication by the end of like their bachelor degree and still not getting into a PhD program. And that's because of many different reasons. They look at different holistic aspects, such as your work ethic, whether you're a good fit for the program, and also just like leadership in general, which I'll just like briefly talk over a little. In terms of funding, as I mentioned before, a good program will cover for everything. So they cover your tuition fees, your insurance, and also give you an annual stipend for you to live on, because essentially you're working for the lab as well. So if you do get into programs that does not cover for your tuition or giving you a stipend, that's kind of a red flag. And that's because it means that the lab requires your money to kind of keep running the lab, and which is not the case that you want, because you want a lab to kind of have its own grants and fundings. As I mentioned before, leadership and diversity is really important in your application. You want to show that, and that's because the admission committee are just looking for leaders of the future, and also just like advocates for diversity in STEM, and not just the brightest person in the future. And then last but not least, recommendation letters. They are so important because they give the admission committee a chance to look at you from a different perspective. So that's why you really need to build your relationship with your potential recommenders really early on. For me, for instance, I built them as soon as I started my research work at the end of the first year. And the second year, just really get to tell them like, hey, I might be thinking of applying to PhD programs. By then, like, if you could give me a good recommendation, that'd be really appreciated. And then always maintain a good relationship throughout your recommenders. So even though you have quit the lab, just checking in on them, just saying like, hey, how's the project going on? How's the lab coming about? Just to kind of maintain that connection is really key, because when it's time for you to ask for a letter recommendation from them, they can remember like what you have contributed, and yeah, basically talk about the good things that you have. And then that leads to the next point. You always want to ask for a strong letter of recommendation. You rather get a strong letter of recommendation from, let's say, like an early stage professor, rather than a weak letter recommendation that says that, oh, he barely does this work, but he makes it by and stuff like this, from a really like well-known professor, because that's going to hurt your application. And then just lastly, just making it really easy for your recommenders to kind of like navigate, because you'll be applying to around like 8 to 10 different PhD programs, all with different links to submit the recommendation letters, all with different application deadline. So usually you just want to keep it easy by like making a spreadsheet so that they can just look at it really easy. And my biggest tip as well is really to start everything early on. That's something that I wish I would have done for myself as well. Just start early into researching the schools that you want, starting early to build that relationship with your recommenders. Start early if you can in your research experience and whatnot as well. Yeah, so thank you for listening to my spiel about PhD programs in the US, and also just about my life here as a first-year PhD student. Yeah, I'm glad to take any questions.